In Chapter 24, we're going to take a look at the mechanisms responsible for species evolution, as well as those responsible for the preservation of species once established. As previously discussed, microevolution is a process that involves changes within a population's gene pool due to migrations, mutation, natural selection, genetic drift, and non-random mating. Over time, the cumulative effects of these changes within the gene pool will lead to large-scale phenotypic changes, leading to speciation, a process known as macroevolution. As we will see, a population is considered to be a separate species once its members can no longer interbreed with those of the parent population and produce viable or fertile offspring. As we will see, a population is considered to be a separate species once its members can no longer interbreed with those of the parent population and produce viable or fertile offspring. To illustrate this point, we can examine the mating between a donkey and a horse. While these individuals are physically capable of interbreeding, the resulting hybrid offspring, a mule, is sterile, mostly due to the failure of its chromosomes to successfully synapse during gamete formation. Therefore, we can consider donkeys and horses separate and distinct species. The microevolutionary forces responsible for the establishment of new species can occur under two distinct mechanisms. Allopatric speciation describes a process whereby a new species arises in a region geographically distinct from the original parent population. Conversely, Sympatric speciation involves a process by which a new species arises in the same geographic region as the original parent species. Allopatric speciation is perhaps the most common mechanism by which new species arise. Allopatric speciation involves the fragmentation of the parent population into two subpopulations, either by way of various geologic processes, such as mountain building, or by way of the founder effect, during which a small group breaks off of the original population and migrates elsewhere. The resulting subpopulations will likely remain in isolation by some formidable geographic barrier. As a result, the allele frequencies within each subpopulation are initially altered by way of genetic drift, whereby certain alleles will be overrepresented, certain other alleles will be underrepresented, or not represented at all. Each of these subpopulations is now subjected to different selecting agents across this geographic barrier. It is expected that their respective gene pools will continue to diverge over time. In other words, we would expect the gene pools of these subpopulations to become more dissimilar as time goes on. A lack of gene flow between these subpopulations, due to the presence of this geographic barrier, will keep these subpopulations genetically distinct from one another until such time that even if they do reunite, there will likely be so many genetic differences between them that they will no longer be able to reproduce and give rise to viable offspring. At such time, these two populations will be considered separate and distinct species. A common example used to illustrate allopatric speciation involves squirrel populations found along the north and the south rim of the Grand Canyon. It is thought that these two species of antelope squirrel were descended from a common ancestor whose population became reproductively isolated across the Colorado River, representing a formidable geographic barrier to gene flow that would otherwise keep these two populations genetically similar. Over time, genetic drift, natural selection, mutation, and other microevolutionary forces altered the gene pools of these two populations to the point of speciation. An adaptive radiation is a form of allopatric speciation that is most commonly observed on islands, where the biodiversity tends to be low simply because islands are initially hard to populate. The low amount of biodiversity common on islands provides a large array of available ecological niches that a founding species can potentially exploit. As members of the founding population begin to move into different ecological niches, they become separated from one another, and therefore they do not mate with one another as often. This, of course, prevents the gene flow that would otherwise keep these groups genetically similar. As a result, these groups become more and more genetically distinct from one another over time to the point of speciation. That Darwin observed throughout the Galapagos Islands, particularly the finch species, are the result of rapid speciation by way of adaptive radiation. That Darwin observed throughout the Galapagos Islands, particularly the finch species, are the result of rapid speciation by way of adaptive radiation. From a single common ancestor, 13 distinct species of finch arose as members of the ancestral population began to occupy separate ecological niches, which resulted in reproductive isolation to the point where each subpopulation became so genetically distinct that it gave rise to separate and distinct species. In contrast to allopatric speciation, sympatric speciation involves the establishment of new species within the same geographic region as the original parent population. Reproductive isolation does not occur by way of some geographic barrier, but rather genetic changes within an individual's genome that causes them to become so genetically distinct from other individuals that they become reproductively isolated, and this may potentially set the stage for the establishment of a new species. Speciation is most common among plants. We can take a look at the following example whereby a particular plant species has a diploid number of 6. If we can imagine a mitotic event in which every single chromosome undergoes non-disjunction, some of the cells of this plant would therefore become tetraploid. If some of these cells are involved in meiosis, they in turn would give rise to diploid gametes. During self-fertilization, these gametes could join to form a tetraploid zygote, giving rise to a plant that is entirely 4N. Such an individual is referred to as an autopolyploid, simply because all of their chromosomes were derived from the same species. Self-fertilization, this autopolyploid individual will give rise to other individuals that are also autopolyploid. However, these individuals could no longer interbreed with those of the original population because of incompatible chromosome numbers. Therefore, these autopolyploid individuals would constitute a separate and distinct species. In this particular example, the offspring was formed in much the same way. However, the gametes, and therefore their chromosomes, were derived from two different species, creating what is often referred to as an allopolyploid individual. Like before, this allopolyploid individual cannot reproduce with members of either parental species because of incompatible chromosome numbers. However, through self-fertilization, such an individual can give rise to a population of individuals just like it. Therefore, this allopolyploid population would again represent a separate and distinct species.